Hi, my name is Melissa Eric, and my presentation is on beer, ancient origins, and new sour frontiers. The tradition of brewing beer has been around since the beginning of civilization. In recent years, the craft brewing industry has exploded with popularity. Consumers are looking for more flavor and more variety in their beer selections, and many brewers have responded to these desires. One style of beer that has been gaining popularity in the past few years is sour beer. While this style is just gaining popularity among craft beer drinkers and brewers, the style has a long history. The origins of fermentation in alcoholic beverages likely predate written history, and its origins are unknown. The first instances of fermentation likely occurred by chance, possibly the result of fruit rotting, honey going stale, or some other spontaneous cause. When studying beer's history, however, the widely accepted guidelines that define beer describe a grain-fermented beverage produced starting from the beginning of agriculture during the Neolithic period to the modern day. With population growth and the establishment of permanent settlements across the globe came the domestication of plants such as einkorn, wheat, and barley, which could be used for fermentation. In order for fermentation to occur, the starch needs to be converted into sugar, and there needs to be yeast present to convert these sugars and other carbohydrates into ethanol. Initially, brewers had some difficulties converting the grains into fermentable sugars, so other sources of sugar were usually added, either in the form of juices or honey. The earliest evidence of beer is in Egypt and dates back to 3500 BC, where excavators found remains of large uncovered vats which were likely used as a brewery. Around the 8th century AD, along with the spread of Christianity throughout northern Europe, beer began to be brewed in monasteries. While monasteries previously mainly produced wine, the cooler climates of northern Europe did not permit the growing of grapes and were rather more friendly to growing barley used for beer. Monks mainly brewed the beer for their own consumption, as water was often contaminated and the beer was safer to drink. However, monastery pubs eventually opened where the beer was sold to the public. The next very crucial step in the evolution of beer brewing was the addition of hops. Hops impart a unique flavor to beer and also aid in its preservation, allowing beer to be transported much longer distances. Demand for beer continued to grow into the late 14th and 15th centuries as towns were growing and there was an increased demand for inns and taverns by traveling merchants. Some other advancements in brewing included the steam engine, refrigeration, glass bottles, and eventually in the 20th century, metal cans. In addition to these processing advancements, a new method of brewing was also discovered. This processing method, known as lagering, was a bottom fermenting process by which slow fermenting yeasts that sink to the bottom are used, as opposed to the top fermenting yeasts previously used in ale brewing. These lager beers were stored longer, which led to a clearer and brighter end product. In modern times, most beers are brewed using the same 4 ingredients: malted barley, water, hops, and yeast. In general, beer production follows one of 2 brewing processes, depending on whether the beer is a top fermented ale or bottom fermented lager. The first step in the brewing process after harvesting the barley is the malting process. Barley must first be malted in order to be used for fermentation, and this is done by steeping the raw barley in water. The water then enters the embryo, which triggers the production of hormones. These hormones are sent to the tissue that surround the starchy endosperm, known as the alurone. The hormones in the alurone then triggered the production of enzymes that break down the cell walls in the alurone and then digest the walls and some of the proteins within the endosperm. The malting process is then completed by heating the steeping barley to stop the germination and to remove any water until the moisture level is below 5%. Following the steeping and kilning is the milling of the malt. Milling breaks down the malt into small enough particles that will enable water to have access easily. The milled malt, or grist, is then moved into a mash mixer or mash tun, where it is mixed with warm water, approximately 45 to 50 degrees Celsius, to induce hydrolysis. During this process, the enzymes that were released during germination process continue to break down any cell wall polysaccharides that were not previously broken down during the malting process. The temperature is then raised to approximately 65 degrees Celsius, at which point the starch is gelatinized. After the mash is held at this temperature, usually for around an hour, the temperature is increased again, this time to 76 degrees Celsius, to stop any enzymatic activity and decrease viscosity. The resulting sugar-water solution is known as wort. After the production of wort, the liquid is moved into a container called the lauter tun, in which the liquid wort is separated from the spent grain. Following the separation of the liquid from the spent grain, the now clear solution flows into a tank called the kettle, where hops are added and it is boiled. This boiling process releases aroma and flavor compounds from the hops, as well as concentrates the worts, inactivates surviving enzymes, sterilizes the wort, and inhibits off flavors. The boiled wort is then sent to a whirlpool, where the centripetal forces drive undissolved solids to the bottom, leaving bright wort on the top. The wort then passes through a heat exchanger in order to be cooled down, and finally flows into a fermentation tank, where oxygen and yeast are added. The yeast ferments the glucose, maltose, sucrose, maltotriose within the wort into ethanol. At this time, enzymes also produce various flavor compounds such as alcohols, esters, fatty acids, and sulfur-containing compounds. The entire fermentation process can take anywhere from 2 to 3 days for a traditional ale and more than 2 weeks for a lager. After fermentation, the beer is further filtered and clarified, stabilizers may be added, and then the beer is generally moved to a bright tank until it is packaged into either can, bottle, keg, cask, or bulk packages. Beginning in the 1990s, there was an explosion of craft breweries, especially in America, and many of them began experimenting with both old and new styles of brewing. One beer to emerge and gain popularity is a style known as sour beer. Sour beers are derived from a variety of old styles of beer, including lambic, gueuze, Berliner Weiss, and other similar beers. These are called mixed fermentation beers, or beers produced with a combination of Saccharomyces, Brettanomyces, Lactobacillus, and Pediococcus, or other less conventional microorganisms for fermentation. One of the oldest surviving styles of sour beer is the lambic style, originating from Belgium. The lambic style is a result of spontaneous fermentation, in which fermentation takes place when beers are fermented with wild yeasts from either an open window of the inside of a barrel or any other multitude of sources. In the case of lambic beers, they are brewed in the Seine River Valley near Brussels, and the wort is infected with the microbes that are present in the air around the river valley, which give the lambics their unique flavor. The inoculated wort is placed inside wooden casks and matured for 1 to 3 years. The resulting product is used as the base for lambic and gueuze beers. The fermentation process for lambic beers actually includes 4 phases. These phases are the Enterobacteriaceae phase, the main fermentation phase, the acidification phase, and the maturation phase. The phases are defined by the microorganisms that are active during each phase. The maturation phase, the final phase, can take anywhere from 1 to 3 years. A gueuze beer is the result of the blending of lambic beers of varying ages together and bottling, where they undergo an additional fermentation step inside the bottles. Another sour style originating in Belgium is the Flanders red or brown style. These beers, brewed in the Flanders region of Belgium, were originally brewed in a style similar to lambics, where the wort would be cooled in open trays and aged inside wooden casks. Another traditional sour beer style is the Berliner Weiss beer, historically produced in Germany. In the traditional production of Berliner Weiss beers, the wort was not boiled, and therefore the lactobacillus that was living on the malt was able to survive and sour the beer later on in the process in the fermenter. In modern times, however, lactobacillus is pitched into the wort along with the yeast after boiling. While these styles of brewing largely fell out of popularity in favor of the consistency of single-strain brews, mixed fermentation beers still thrived in certain areas, and the style was revived around the mid-1990s. In modern times, the production of sour beer is usually similar to the production of other, more common styles. Production is generally the same, with the mixing of the crushed grains with hot water, boiling, adding hops, and chilling. The main difference is the pitching of the wort with Saccharomyces, Lactobacillus, Brettanomyces, Pediococcus, or some combination of them. Many sour beers also utilize unmalted grains such as wheat. This process is unique to sour brewing, as the unmalted grains contribute starch and dextrins. These chains of sugar molecules that make up dextrin are too long for Saccharomyces to break down, but they are easily fermentable by the other microbes used in mixed fermentation beers. There are in fact 2 methods for producing sour beer. The first method is known as kettle souring and is a faster way in which to sour a beer. In kettle souring, the wort is produced in the brew kettle with either no or low amounts of hops, and is then cooled after boiling to around 40 to 45 degrees Celsius. At this point, the wort is inoculated with lactic acid bacteria, such as lactobacillus or pediococcus. This temperature allows the bacteria to grow and produce lactic acid until the pH reaches the desirable level, at which point it is boiled to inactivate the bacteria. This method allows a faster turnaround time and better control over acid production, and is beneficial for small breweries where cross-contamination between beers is an issue and there is not as much storage space. The other, more traditional method of souring is through mixed culture fermentation. In this type of souring, the hopped wort is sent to the fermenter without adding any bacteria first. Once in the fermenter, it is inoculated with either Saccharomyces cerevisiae, or in the case of 100% Brett beers, Brettanomyces. Lactic acid bacteria can be added at this point as well, so the beer can be soured concurrently with the primary fermentation. After the primary fermentation, the beer is transferred to barrels where it is aged for months to years. It is during this stage where the lactic acid bacteria will lower the pH and add distinct flavors. This method of souring produces more complex flavors, but it suffers from drawbacks as well. A brewery must have the space to store these sour barrels, and there is a significant time lag from brewing until the beer is ready for consumption. Additionally, since the microorganisms survive throughout the entire aging process, there is a greater concern for contamination to other so-called clean beers. One of the most common microbes used in mixed fermentation, alongside traditional brewer's yeast, as I've mentioned before, is Brettanomyces. The main species used for brewing are Brettanomyces bruxellensis and Brettanomyces anomalus. Although Brettanomyces is commonly known as a spoilage organism, causing off notes described as barnyard, medicinal, leathery, and other descriptors, if used properly, the organism can produce many more pleasurable flavors, such as pineapple, mango, pear, and grape. The other microorganism utilized in the production of sour beer is bacteria in the genus Lactobacillus. Traditionally, the German beer style Gose and Berliner Weiss use Lactobacillus as their primary souring microbe. Typically, Lactobacillus strains cannot survive in a high IBU environment due to hop acids, and therefore it is imperative that brewers who are intending to sour their beers with Lactobacillus choose resistant strains or add the hops after it undergoes fermentation. Lactic acid bacteria in the genus Pediococcus can also be used for the production of sour beers. The most commonly used species for brewing is Pediococcus damnosus, as it is the most tolerant to high acid conditions. One microorganism gaining attention for its ability to produce sour beers through primary souring is the yeast L. thermotolerans. Previously, this yeast was used mainly for the production of wine, but has recently been studied more and more in the production of sour beer, due to its proficiency in producing lactic acid. It is shown to increase the production of ethyl lactate as well, which gives off a fruity aroma, which can be beneficial for a sour beer. An additional benefit of using L. thermotolerans in sour beer is that it is able to survive at higher alcohol content than other microorganisms, as far as up to 9% alcohol by volume. With the growing popularity of craft beer, many brewers are looking for a bit more diversity in the microbes that they use. Some new yeasts that have become of interest to brewers include Saccharomyces eubayanus, Torulaspora delbrecki, and Maracchia gelida. These yeasts have the potential to impart novel flavors, produce lower alcohol beers, and conduct primary souring. Along with the new interest in using novel and wild yeast to ferment sour beers is the interest in the functionality of the microorganisms used to ferment the beers. While at the present time there are no beers being commercialized with probiotic bacterium, there has been some research into the topic. So, in conclusion, sour beers is really a growing market, and there's a lot of renewed interest in this area, and a lot of scientists are currently conducting studies to find new and innovative bacteria and yeasts to use to create sour beers. Thank you.